What I want to talk about more is the biology, how this happens. So this is cancer treatment is a really cool example of where research has helped treatment. For a long time it didn't, but now recently that's coming true. So basically the idea is if you understand how it works under normal circumstances, how cells divide, then it turns out understanding that has really helped to understand what goes wrong in cancer. So let me show you. We'll start with how it should work and then how it can go wrong. So growth control. Like I said, cells only divide when told to. And so basically, if you've got a cell, on its surface are a whole bunch of receptor molecules. And these are receptor proteins for different growth factors. And these are different on different cell types. So every cell's got receptors all over the surface. Every cell needs to be able to trigger to grow. They're different on different cells. How is that possible? The genes for those receptors are the same in every cell. Some genes are turned on in others. So like the cells in your bone marrow, which are going to give rise to red blood cells, they express the gene for the erythropoietin receptor. So when erythropoietin comes along, they'll get stimulated. Cells in my brain don't have the erythropoietin receptor because they don't need to respond to erythropoietin. They don't make red blood cells. So every cell has this particular collection of receptors on its surface. And there are a whole set, hundreds of different growth factors. And these are made by other cells. And so what I want to do is get ever sort of increasingly fine focus. So we'll do sort of big picture, and then we'll come back again, finer details, finer details, finer details. We'll get more details on Wednesday, but I want to give you the big picture today. So how do these growth factors work? And the idea is, if you've got a cell, and you got the nucleus in the middle, if the right combination of growth factors are present, they bind to the receptors. So like there's some receptors on the surface, and if the circle one and the square one are around, they both send their message to the nucleus. Then the receptors send chemical messages to the genes in the nucleus, and there are multiple interconnected message pathways. And then 3 in the nucleus. I'll explain this all in a second. In the nucleus, certain genes are activated for transcription, and then translation, and then the protein's made, and for the new proteins are the machinery for cell division. So like DNA polymerase, etc. And number 5, the cell divides. So the idea is that it's one of my more atrocious 5s. All right. Right, growth factors. So remember, in the end, growth factor comes, cell divides. Let's break it down a little bit more. In the end, if the cell is going to divide, it needs certain proteins. It's going to need DNA polymerase to replicate DNA. It's going to need a bunch of other things to get the cell divided in half, split in half, all that kind of stuff. Those genes are going to need to get transcribed. So in the end, the way it works is growth factors are present. The receptors send a chemical message, and it turns out to be a really complicated set of pathways. We'll talk about some of them. Send a message to the nucleus that says, transcribe these 25, 35 genes. They get transcribed. RNA gets translated. Protein gets made. Now the cell has the equipment to divide. When the equipment is present, it does its job, and the cell divides. So we're going to break these down in more detail over the next couple of lectures. Questions about that process before we talk about a little bit more detail?